Okay, everybody. So, Assalamu alaikum, everyone. I'm repeating this entire lecture for you all again, okay? So, if you have missed out anything, this is a good thing for you all to uh, learn that stuff, okay? So, this lecture is based on how exactly the dose produces its effect by combining with the receptor, okay? And how exactly the doses are being monitored and everything, okay? So, let's start. Factors contributing to drug effect. All right. So the first thing that comes up in our mind, as I said in the first lecture, it depends on how many receptors are there within the cell membrane. Okay. So you see, if you look over here, okay, so receptors may be differentially expressed in one tissue to the next and therefore mediate different levels of biological responses. What do I mean by that? Let's elaborate it with the help of the example, okay? That, for example, in the heart, okay, beta 1 receptors are there in abundant amount, okay? But in the lungs, bronchioles, and arteries of the skeletal muscles, okay, we have beta 2 receptors in abundant amount. Now, let's just say if I am taking beta 1 receptor, okay, so I will, so the effect would be produced in the heart and not in the lung. Why not in the lung? I told you all in the first lecture that agonist means something that complements the function. For example, if I am taking a medicine that is going to enhance the function of norepinephrine in my body, okay? So that means that is the agonist drug, okay, which is promoting the effect of norepinephrine, okay? Maybe I'm too sluggish, that's why I'm taking it, okay? But for example, if I take an antagonist drug, okay, just in case, maybe I'm hyperactive, okay? So somebody gave me a drug which is norepinephrine antagonist, okay? Antagonist is when it blocks the effect, okay, or diminishes the effect, all right? So when it's going to block the effect, so that's going to be called antagonist. And let's say if I want not 100% blockage and I want, let's say 50% blockage and 50% like I want it to work, okay? So I'll give it partial agonist, okay? So over here, we are concentrating on the beta 1 agonist drug, okay? So beta 1 agonist drug would produce effect on my heart and not on the lung or skeletal muscles. Similarly, beta 2 receptors, okay, would produce effect on the skeletal muscles and the lungs instead of producing a huge impact on the heart, okay? So you need to remember how exactly these receptors work and where exactly they are in the abundant amount. I'll share a table with you in the next few classes, okay? All right. Okay. Now, signal amplification. We have already talked about it, that how exactly signal amplification happens. What happens in the signal amplification? A ligand gets attached, okay, and then an immense response is produced, a cascade of events is produced, all right? Now, why exactly this is happening? Like signal amplification, this means that it could be done by one way. That is, you take a medicine which initiates an events, like a number of events. Okay, this is one way. The other way is that, for example, you have a spare receptor. Okay, so you don't need to worry about it. You just take the drug in high amount. Okay, and it will start to dock on the spare receptors and to produce effect. Now, what are the spare receptors? Let's talk about it in detail. For example, you have 10 chairs in your home, okay, and you have 4 people living in the house, all right? So how many chairs are spare? There are 6 chairs spare in your house, right? Similarly, if you would relate the chairs to the receptors, you would easily understand what I meant to say was that, let's just say your, for example, here I have jotted down example of glucose molecule, okay? So for example, I had initially hundreds of 100, let's say 100, not 100. Yeah, over here also I have written 100. Okay, so let's say I had 100 of no. Okay, so let's say I had 100 of receptors, okay, in my cells. All right, and in the normal body function I needed only, let's say, 20 or maybe 10, not much. Okay, but all of a sudden I started working really really hard, and my body needed an excess amount of energy. So how would my body support it? My body was supported by, taking in a lot of glucose, okay, and utilizing it for cellular respiration. Now, how exactly this would happen? This would happen, a huge amount of glucose intake would happen because of the spare receptors that I have, okay? The extra receptors that I had in my body, which were not used in the normal body functions, all right? So, when we talk about insulin, okay, so we have almost 99% its pair receptors by which it can bind. And when we talk about human heart, okay, so we have almost 5 to 10% of the beta adrenoreceptors. And when I say 5 to 10%, it means they are not that much, they are a bit less, okay? And that is why you need to be very much careful when you are, dealing with this specific organ of the body. Okay, wait. All right. Now comes up some definitions which I want you all to learn, and we will talk about them more in the depth. All right. Okay. So the first one is receptor down regulation. Then is desensitization. Then is receptor upregulation. Then is supersensitivity. So what is that? Let's talk about it. Okay. Remember, in my very first video, I talked to you about that. According to the new theories, it is no more like that. That there was a lock and key model in which substrate was there and product was there, which was produced, and the enzyme got attached with the molecule, and it was either broken up or built up into a new thing. The concept is no more like that. Okay, the according to the new concept is like that there is you can say, um, there is a shift in between active receptors and inactive receptors. Okay, we represent these by R and R steric. All right, so the number of activated receptors actually tell us that how much of the effect can be produced by the drug that we are taking. For example, if you look at the very first thing, okay, that is receptor down regulation. When you hear the word down regulation, it means it already comes into your mind that there is something which is diminishing, which is decreasing, right? So a decrease in the total number of target cell receptors for a given messenger or ligand in response to chronic high extracellular concentration of the messenger. Wait, what do I mean by that? Over here, chronic high concentration was there of the messenger, okay, due to which the receptors got decreased, okay, because of the messenger. Sorry guys, because of the excess amount of the messengers or ligands, okay, the receptors got decreased. Okay, and now what is desensitization? This is again a very important concept. The receptor becomes desensitized as it becomes unresponsive to the ligand due to continuous stimulation. For example, I am taking a medicine on daily basis, okay, and now my receptors are getting desensitized. They need an extra amount, okay. Agonist administration results in molecular changes of the receptors such that the receptor undergoes endocytosis and is sequestered within the cell, unavailable for further agonist interaction. It means that this receptor, okay, it will actually get detached from the cell membrane and it will get inside. Okay, we are looking over here the terminology endocytosis. Okay, it means that the receptor which was attached on the cell membrane, okay, now it is getting detached from there and it's getting inside the cell membrane and it is becoming unavailable for binding. Okay, so the number of these receptors are regained after some time. Okay, but then again after some time. Okay, and that period in which those receptors are not there, okay, because they were inside the cytoplasm. Okay, so that period, specific period when the cell was unresponsive, okay, that period is called refractory period. Remember we talked about voltage gated ion channels when we talk about how exactly minus 55 threshold, minus 55 millivolts threshold was not activating the neurotransmitter. Okay, it was because of this reason. Okay, that was desensitization when it undergone into the repolarization period. All right. Okay, now is receptor upregulation, as the term indicates to you, upregulation. Okay, it means that the concentration is getting increased. Okay, now an increase in the number of target cell receptor for a given messenger in response to chronic low extracellular concentration of the ligand. Over here, because of the excess ligand, the receptors got increased, but over here, the receptors got increased due to the less number of the ligands, right? Then is supersensitivity, the increased responsiveness of a target cell to a given messenger ligand resulting from receptor upregulation. Okay, so the receptor gets extra sensitive for that particular ligand and the effect is produced like that. All right. Okay, so talking about this tachyphylaxis. Sorry. Okay, now what is tachyphylaxis? Tachy, okay, you will hear the word tacky like joined up with so much many other things. Okay, so many other words. So this tacky means fast. Okay, so when I say tachyphylaxis, okay, it means that it is acute tolerance. All right, it is fast occurring tolerance. Okay, over here I have written names of common drugs which produce tachyphylaxis. Okay, you have to memorize these names. Okay, that is nitroglycerin, amphetamine, ephedrine, dobutamine, SSRIs, okay, TCAs. All right, so these are the drugs. Okay, by the way, SSRIs and TCA, it's a huge class of drugs. Okay, it's not one drug. Okay, it's a huge class of drugs which we'll be studying inshallah. Okay, now tachyphylaxis is used to describe desensitization that occurs very rapidly due to repeated administration of the drug in a short time. For example, tyramine. For let's say I started taking a medicine again and again, and now no matter either I take small amount of that medicine or I start to take a huge amount of that medicine, okay, my receptors will not produce a response at all. Okay, so this can occur without a change in the number of receptors. Okay, it's just that the receptor is saying, okay, I'm no more functioning. Okay, and it is not dependent on the number of receptors. All right, it's just this, that the receptors have totally quit it. Okay, or the ability of the receptor to affect the downstream signaling molecules. Increasing the dose of the drug will not improve the response. Like I said, that since the receptors have given up, so now it doesn't matter either you increase or decrease the drug concentration. All right, then is tolerance. Okay, tolerance and tachyphylaxis, okay, they are very frequently tested if students know the difference between them. Okay, because they're almost alike. All right, in tachyphylaxis, I've told you that the response is diminished at all. Okay. In tolerance, okay, it happens gradually. Okay. So what is the difference? In tachyphylaxis, it's like super fast. In tolerance, it's like slow response. Okay. Slow response to a drug that occurs. Oh, wow. It's okay. My this lecture is getting recorded. Okay, I'll upload that for you on the YouTube. Okay, so you don't have to worry about. it at all. Okay, all right. So those who are getting signals, okay, I'm delivering this lecture to you online right now. Okay, all right. So tolerance is conventionally used to describe a more gradual loss of response to a drug that occurs over days or weeks. All right, so it takes days and weeks to produce tolerance. Okay, so now everybody in the chat box, okay, I want you all to tell me that which one is tachyphylaxis? Which one do you think? Graph 1 or graph 2? In the chat box, give me the response. Okay. I hope my recording is resumed. Yeah. Okay. So let's see. Okay. So guys, this one here is tachyphylaxis, okay? And this one here is tolerance, okay? So just do wait. Oh no, oh no, oh no. Okay. So just to confuse you people, okay, I actually selected these pictures intentionally, okay, in which there were three graphs, okay. So you see, you give one time the medicine, okay, dose, it produces the maximum response and then decreased. Then you give again it's decreased, okay, because then your brain remembers, okay, that how exactly it works. Okay, I'm going to wait. Oh no. I don't know why today my computer is not responding the way I want it. Okay. So everybody, you all saw the what exactly, how exactly the tolerance is, producing a bad impact in our body. Okay. Yes, in tachyphylaxis, okay, the drug is no more recognized at all. But in tolerance, in order to achieve the same peak level, okay. So, let's say over here, wait, let me move my screen. Okay, you see, dose administration over time, okay. So you see, you have to increase the dose administration in order to achieve the same peak level, okay. Let's say if your dose, okay, you're not increasing, okay, and it's the same, all right, so it's going to keep on diminishing, okay. That is why you see, there's some medicines, okay, that is opioid, of course. For example, Vitacon, okay. So what it does is this, that it produces dependence, addiction, okay. And that's why some people even start to take, a dosage of a week in one day only, all right. And that's how a lot of people die because of this drug tolerance developed in your body, okay. All right. So now we are coming up to our next topic, not topic like subtopic, okay, which is efficacy versus potency. What is efficacy and what is potency? First of all, let me crack these words for you, okay? For example, we are discussing, okay, and we are saying that for COVID-19, okay, I had a drug A and it really worked for me. And the other person says that, yeah, drug A was good, but drug B was superb. It treated me like in one day only, okay? So now you get it? Efficacy means over here, you see, refers to the relative ability of a drug receptor complex to produce a maximum functional response. Okay, so this means efficacy means how much benefit a drug is giving you. All right, potency. What is potency? Potency means that how much of the drug, how much how much quantity of the drug actually helped you to get the response. For example, when I develop a headache, okay, sometimes I take one Panadol, all right, and sometimes I have to take two Panadols. I have to increase its potency because of the headache that I have. It's unbearable. Clear, everybody? Uh, I'm not able to respond to you. Okay, I'll respond to you afterwards. Okay. All right. Then is, you see over here, I have jotted down an example for you. That is candesartan and irbesartan are angiotensin receptor blockers that are used alone or in combination to treat hypertension. Now, this drug, you see, candesartan is more potent, okay, than irbesartan because the dose range for this is 4 to 32. It means that in just 4 to 32, it produces effect, okay, as compared to the other one which was 75 to 300 milligram, okay. So this is the difference. You see why? Okay, so it started to produce effect at a higher dose, okay, and the other one, okay, the this is the difference, okay, in between potency. And when we talk about efficacy, we talk about that which drug produce the maximum of the response, okay, how much it benefited. So potency denotes to the amount of the drug needed to produce a given effect, okay. uh, all right, now comes up to quantal dose response, okay. What is quantal? Quantal tells you magnitude of dose on the proportion of a population that responds, okay. So it means that it is a study which we do on a specific population, and then we try to understand that how much of a dose produce how much response in a specific population, okay. Let's talk more about it. Okay, so this is therapeutic index. Okay, what is therapeutic index? We usually, represent it with the term TI. Okay, and we have two formulas for it. That is TD50 over ED50 or LD50 over ED50. Now, why this discrepancy and what is ED? ED is the median effective dose, okay, which would produce a response, okay, and TD is the toxic dose which we evaluate in humans, okay, or animals, and leader in human, okay, we in humans, okay, we detect the toxic dose, and in animals we detect the lethal dose, okay. Of course, in human being it's not possible for us to detect the lethal dose. We don't want anybody to die, so we don't check it, okay. But in animals we do check the lethal dose, that is LD50. Now there is a mnemonic which I have attached, okay, so that you all can remember the formula. That is style for animals, okay, TILE, okay. So lethal dose divided by effective dose. And for humans it's TED. You know the it's a very famous channel. You all must watch it. I watch it. Okay. So it's TED. Okay. So it's TED. Okay. So this is for humans. Okay. For human, remember this, and for animals, remember this. Now, what exactly is a therapeutic index? You look over here. Let's just say you have a medicine, okay, and technically speaking, if you want to know that, let's say if I have three medicines, okay, so I would ask that which one has the huge therapeutic index, okay. Why? Because let's say if this green portion is narrow, okay, so definitely it's a threat for me. It means if a microgram is given extra or is given in the less amount, okay, so either it's going to produce no response at all or it's going to produce a lethal response, okay. So I don't want to die, so I would take care of the therapeutic index, okay, that how much of the drug has been given to me so that I can be benefited rather than having negative impact of the medicine on me, okay. So let's talk more about it. Okay, so you see over here, when I'm taking the when I've started to take the medicine, okay, the what was 50 is in the 50% of the population, okay, here, okay, the 50, all right. So let's just say in the very start, okay, there was no effect at all, then there is therapeutic effect. And let's say if I increase the dose, okay, then you see these are the log concentrations, okay. So you see a red line is achieved, which means that this is the toxic dose. Now I would, start to experience the toxic effects. So anybody would opt for the dose which produces therapeutic effects on me. Okay. All right. Now, another terminology, therapeutic window. What is that? Therapeutic window or pharmaceutical window of a drug is the range of drug dosage which can treat disease effectively without having toxic effects. Medication with a smaller therapeutic window must be administered with care and control, frequently measuring blood concentration of a drug to avoid any harm. Okay, you see, this is the therapeutic window. This is the duration of action it is producing, and this is the peak effect. Now, let's compare therapeutic window and therapeutic index together. Okay, so you see, the larger the therapeutic window, the safer the drug is. Okay, it means that if it gives us a huge amount of period for us. Okay, so when we are talking about the therapeutic window, okay, you see there are two medicines. Okay, this is medicine A and this is medicine B. Okay, of medicine A, the therapeutic window is huge. Okay, however, of medicine B, the therapeutic window is small. Okay, so A is penicillin. Okay, and B is warfarin, which is used for thinning of the blood. Okay, so it means that warfarin I have to give it very carefully. Okay, I have to take care that no extra amount is being given because it can easily produce a toxic effect. Okay, and there is a huge gap. All right, however, in therapeutic window there is a huge gap. Okay, and I can easily like do some about ups and downs, and it means that I'm not saying that it's a very safe drug and you can like take it just like that, but it's just this that you see this drug would produce effect in a longer amount of time as compared to this drug. Okay, for example, we take Panadol like very frequently. It is a drug which is easily available. However, let's just say somebody takes in a huge amount of Panadol, so definitely the person would experience the toxic effect. All right. Okay, everybody. Thank you so much. Um, Wait, let me end so I can respond.